This presentation is for Olix. Olix is working on nucleic acid RNA to develop new drugs and currently working on macular degeneration or alopecia, among other diseases as well. They conduct topical administration. They're currently working on 10 or so new drugs, and this time around, the presentation will be conducted by President Lee Dong-gi. Hello, good to see you. Currently, you're working on 10 new drugs. Please introduce your company.
So based on our company material, I would like to begin my introduction. So let's begin with the overview of the company. Our Olix is based on our own RNA interference treatment, and it's a company coming up with new developments of drugs. And we were established in 2010, and based on our technology, we enlisted on Costock in July 2018, and it is located in Gwangju, Suwon, and we have 67 staff members. And I'm a founder and entrepreneur of the company, and I was assistant professor at Pohang Technology University and currently a professor at Sungkyunkwan University. And we founded this company thanks to technology transfer. And what we first did at the first stage was establishing a company, a branch in the United States. We established Boston, US, and Boston branch is focusing on business operation as well as clinical trial development. And in San Diego, we're working on compound chemistry, and we established this chemistry lab in there. And let's move on to the growth history. Since the establishment, we had to invest a few years to come up with core technology. And in 2014, we began the first Series A investment, and then we launched our massive development of our technology. And later on, we got approval from Korea's technology in 2016. And in 2018, we enlisted on Costock. And early last year in 2019, we came up with a 301 program of macular degeneration, and we got European patent rights with French company Thea. And in terms of our board members, for clinical trials and drug development has to do with various things. For example, clinical trials, animal tests, and toxicity tests, and clinical trials and tests. We need a lot of experts from different fields. So this slide shows that we have a lot of experts from different fields. And also, nowadays we are currently focusing on especially the ophthalmology and liver disease. So we have recruited these advisors and consultants who provide advice to our company. Let's move on to the core technology. The RNA interference technology is what I have to explain, and as far as you may be fully aware, the DNA genetic information has the RNA, and they process data to this, and when they generate this protein, and if they excessively produce too much protein, then we may have diseases. But the first generation low molecule chemistry and second generation antibody new treatments were like working by producing proteins to deal with these diseases. But the third generation is oligonucleic acid based protein. So this is the special characteristics, and our special treatment is the most effective and efficient one that can be used for this purpose. So the RNA interference technology is based on the third generation oligonucleic acid, and it can approach all the different circumstances, and it can clearly deter the protein generation. And the human body's DNA has about 22,000 DNA data pieces, and the targets were untouchable, unapproachable by the first and second generation, so they are called undruggable targets. But because of the DNA differences, if we make use of the oligonucleic acid, then we can target all the undruggable targets, and we can deliver these drugs. And especially the RNA interference technology is the most effective RNA controlling method. And as shown from the slide, there are two spiral-shaped string traits, and if we deliver this into target, then we can deter the massive excessive generation of protein and deal with this. The RNA technology was firstly defined in 1998, and in 2006 it won the Nobel Prize in Physiology or Medicine. And all the undruggable targets that couldn't be defined are now defined thanks to the third generation. And based on this one platform, we can come up with a new drug in a very short, limited time. So this RNA interference treatment is gaining a lot of attention. And in 2018, it got approval from the US FDA, and in the same year later, got the second approval. So nowadays, we expect a lot of new RNA-based interference treatment coming up in the market. So let's move on to the RNA interference technology limitations. So I'll have to begin with the challenges in the conventional years. So those early years RNA interference technology was this formed with this siRNA, which was symmetric. And if we used this siRNA, which are symmetric, then we came up with a lot of side effects which we didn't intend. They deterred the normal generation of protein, or there were a lot of interference, and it deterred normal activation. And when we injected this into eyes, then it could hurt retina cells were reported. And besides all the side effects, the siRNA, the acid stem cell, how are we going to clearly, successfully deliver this? Was the another issue. So we Olix thought about this conventional RNA interference challenges, and we wanted to overcome, and we came up with this technology. So this siRNA or cp-asiRNA technology came up by our company, could overcome the delivery issue, and we could mitigate those various side effects. So it's a platform for that. And as for the cp-asiRNA, there is this supporting attachment to the cell, so we can penetrate the barrier of the cell and administer this dosage. So we can use this for topical administration. And recently, nowadays, in terms of RNA interference, there are a lot of companies are having this trend of using the GalNAc chemistry cell to target specific cells so they can come up with a treatment for liver diseases. So Olix is also working on this GalNAc chemistry, so we attach this to successfully and effectively deliver this nucleic acid to our target to come up with a potential new drug. The current status quo of the global development of the RNA treatment is shown from here. So this is what happened in recent 2 to 3 years. And if you take a look at the slide, there are more than 14 big deals which are worth 200 billion Korean won, or more than 7 mega deals which are worth 1 trillion Korean won. So, namely, there are a lot of gaining attention into this field. Next is our platform based growth potential. It shows our growth potentials and expansionality. So I'll elaborate about this strategy. So platform, I am keep using this term platform. What I mean by this is that the structure of nucleic acid is almost all set, just like shown from this slide. And what matters is what kind of target we set and what kind of target DNA we find out, and if we alter that. There are nucleic sequences, which are the alphabetical system of DNA, and if we successfully alter the nucleic sequence, then we can come up with a very successful candidate for a new drug. And usually it takes about 3 months to come up with a candidate, and compared to the first generation and second generation, we can clearly decrease the time for trials. And as I have just mentioned, there are de-druggable targets which can't be approached by the first and second generation, but by using the third generation, we can come up with various pipelines with very limited time. So in the early stage, we can conduct technology transfer, or we can come up with various pipelines. So we have this business-related strength, and that is all thanks to the platform that is possessed by Olix. So in terms of the RNA interference treatment platform, we can deal with various diseases. So for skin, we can come up with hypertrophic scar treatment, and for eye, we can come up with macular degeneration or retina-related diseases. And for lung, we are working on idiopathic pulmonary fibrosis, and also we're working on the coronavirus disease treatment. And for others, there are nervous system or liver disease or immuno-anti-cancer therapies, among others. So let's move on to the core programs. The first cure is the Olix 101A, which covers the hypertrophic scar. So when we have a very big scar, or if we go through a C-section or a thyroid gland surgery, then our skins have to heal up and they generate collagen or fiber. And if we generate too much collagen or fiber, then we create this hypertrophic scar. And 1 or 2% of the human population have this K-braid disease, which is like our skin heal up like a tumor. But currently, for these diseases, there is no approved treatment by the FDA. So we make use of the CTGF, the targeted protein treatment, is under trials, and by deterring the fibrosis of it to cure this. And currently in Korea, we are conducting the second clinical trial, and the Hugel company has the Asian rights. So under the leadership of Hugel, we are currently working on the second trial, and also we, on by own, conducted the first clinical trial in the UK, and we're planning to expand to the US clinical trial. Now let's move on to the age-related macular degeneration. What is age-related macular degeneration? This is a very common disease for the elderly of 60 or older. So when there is a problem in the yellow spot at the center of the retina, and they go blind because of this, and there are wet and dry age-related macular degeneration. And for wet, Lucentis and Eylea are very effective. However, for dry age-related macular degeneration, there is no clear treatment. So for this dry macular degeneration, we are currently working on developing a new drug. And as far as the retina disease goes more and more, and there are more and more scars, and they cannot avoid going blind because of this dry macular degeneration. And there is no clear treatment right now. So we are currently working on this dry macular degeneration as well as retina fibrosis treatment. The OLX 301A is can be worked on both wet and dry, and the images shown below there on the left is animal test for rat, and when we administered our dosage is the far right on the chart. When we injected high dose, then the retina was clearly protected. And on the right, we conducted a primate or monkey animal trial. And when we projected laser on the retina of the monkey, then we could see blood leaking. However, if we administered our drug, then it dried the scar. So thanks to this animal test, we could see that this can be used for both wet and dry macular degeneration. And currently we're working on the toxicity test. And the Olix 301A program, in the very early stage, we had a contract with French Thea, which was worth 80.7 billion Korean won worth of amount. Let's move on to the Olix 301D. So this uses the CTGF as a target. So namely, if we block it on the skin, then it can be a skin cure, and if we use it for the retina, then it could be a retina cure. So the red colored one is that we dyed collagen. If you take a look at the second one, you can see the collagen gradually growing, but if you take a look at the right one, then the collagen's volume decreased. So this shows that the retina's fibrosis could be blocked, thankfully. And if you look at the right chart, it has a clear efficacy on age-related macular degeneration fibrosis, and currently we are also working on the toxicity test. Next, let's move on to lungs. Let's talk about OLX 201A. Lung fibrosis can be easily understood if you think about humidifier germicide. if this germicide gives scars on our lungs, and if we heal up, then lungs, which usually has to be soft, hardens, then within 3 to 5 years of diagnosis, you die. But currently, there is no clear treatment for this. So a lot of pharmaceutical companies are paying a lot of attention to this as well. The Olix 201A is inhale-based dosage treatment, and just like I said, the CTGF, the protein, is the target, and based on inhale-based dosage, we have proved its deterrence power. And in the very early stage, where Olix is working in cooperation with Agency for Science, Technology and Research of Singapore, and Singaporean this research center proves our efficacy. And the bottom blue colored images down there shows the lungs of a mouse, and the more dosage that we administered, there are more holes on the image, which means that thanks to the dosage, we successfully decreased the fibrosis of the mice's lungs. So in terms of the platform-based treatment for lungs, we are currently working on the coronavirus treatment. Of course, we are in the very early stage. However, the coronavirus's genome is formed in the RNA as shown in the bottom. So if we target some specific traits of the coronavirus, then we can deter the coronavirus's RNA successfully, and we are currently working on animal tests. Last is liver disease treatment. Based on the GalNAc platform, we add this nucleic acid on the target to successfully deliver. And we have secured a lot of different platforms. And based on all the platforms, we recently recruited these 3 liver disease specialists from Harvard University as consultants in order to come up with successful target research. So in the second half of this year, based on our specific treatment, I think we can conduct clinical research in the near future. So far, I have explained about the pipelines of our company, and last but not least, the ending slide. I would like to say that Olix is based on the 3rd-generation oligonucleic acid, and we possess this technology. And this, we have successfully began our clinical trial, first time in Asia. So I would like to say that we are the leader in Asia. And based on our platform, we have secured a lot of pipelines so we can come up with various treatments very rapidly. So we have this multiple approach. So based on this stable and fast research development, we can make a great success. Yes, that is it.
Thank you for your presentation. So, for a bio company, technology is the key. So, as far as I understood, your company's key is the technology itself. So, not only the coronavirus, but I'm looking forward to seeing a great success of your development in the future. It was President Lee Dong-gi of